Thank you, and good morning to everybody. It's quite intimidating to follow Prof. Wingfield in the program. And also, it's quite intimidating because I'm not a microbiologist or even a botanist. I'm actually a zoologist by training. But over the last few years, I've been given the responsibility of running 2 initiatives for collections, and so today I'll be talking about them, and I also won't be giving a lot of detailed research data. I just have to get it to change. Okay, so I think I'm going to cover briefly what these 2 initiatives are and what they're trying to do, and then look at how they actually contribute to plant health. And try and look at a value chain, sorry, excuse me, of how these collections are associated with plant health, and then look at some of the challenges that we face. So just an overview of the Natural Science Collections Facility. This is a Department of Science and Innovation initiative. It's part of the South African Research Infrastructure Roadmap, and it was initiated early in 2017. It's a network of institutions that hold natural science collections, and it includes museums, science councils, and 3 university herbaria. And it's coordinated through SANBI here in Pretoria. And it's really focusing on preserved or non-living collections of plants and animals, fossils, and fungi. The Biodiversity Biobanks of South Africa, or BBSA, is another research infrastructure roadmap project that's a little bit slower at getting going than the NSCF, but it's going now. And the scope of this is frozen plant and animal tissue, DNA extracts, gene banks, and cultures, microbial cultures, and it also includes agricultural biodiversity. So agri-biodiversity and relevant collections. And the core biobanks, so those that are participating in the BBSA, are the ones that hold these materials for long-term preservation on behalf of the nation, but then also other samples they make accessible internationally and nationally outside of their own institution for research and development. So just to give you some idea, we haven't finalized agreements with institutions, but participating will be SANBI's own Millennium Seed Bank, which is just getting going here, although it's been going for a long time at Kew. And then also a plant, mostly indigenous plant DNA bank. And then the Department Agriculture have got a plant germplasm collection. It's mostly crops. Then Free State is a yeast collection, Western Cape University microbial cultures, and then the ARC has a whole range of different biobanks that fit into the BBSA. So that's from germplasm tissue culture for crops and fodder, and then also microbial cultures. So what are these 2 initiatives trying to achieve? I think they both, they're separate, but they have some common agendas. So the first is to secure these collections, and that includes rescuing orphan collections and making sure that we comply with the standards set globally or our own standards that we've developed, like for the NSCF, based on international standards. So the first objective is just to make sure that these collections are safe. The second is to upgrade the data sets and the data management systems and make specimen data accessible. And then increase the use of collections for research, development, and decision-making. So really to open up and promote access to these collections. And then also to increase awareness of them, of the collections and their value for a whole range of different purposes. To achieve the objectives, we have to have staff and capacity development. We have to have professional ways of working for the people who look after these collections and who manage the data. And we also have to have transformational change. And that's a really big issue, and I'll come back to it a bit later, but looking at the current situation where there's a lot of people working in isolation and a lot of collections in isolation, a lot of institutions in isolation, and really changing that to being part of a collective. And there's other things that need to change as well. And then we also need to make sure that there is resourcing for these collections. And then we can't do anything unless we can navigate our very complex legislation and permitting requirements. I'll come back to that too. So the natural science collections is a little bit harder to kind of pinpoint how these collections of dead things contribute to plant health. And it's really, when you start to think about it, they are really important. They are a record of what species we've got and where they've been recorded and how this has changed over time. And a lot of these collections span almost 200 years. And so there's lots of historical data that you can map and show how invasive species have expanded their range. Threatened species might have shrunk in their range. You can look at origins of disease or pests. And in addition to that, these collections are really used as a reference for identification of biological materials. And that includes soil organisms, pests, pollinators, indigenous and alien plant species. So all of this is relevant to plant health. The data from these collections, the specimen data, so the locality and when they were collected and what they are, is really critical to doing conservation assessments. And so the Red List of South African Plants, it was published in 2009, but it's updated on an ongoing basis. So the assessment of which plants are threatened and need protection, and then also pollinators like the Red List assessment or the conservation assessment of butterflies. All of that, those assessments, the data comes from these collections. And the data from the collections also feeds into environmental impact assessments and development applications, and also feeds into spatial planning that we use to identify where critical ecosystems are for conservation. And so if we don't have healthy ecosystems, we won't have healthy plants, whether it's crops, whether it's indigenous species, so it's all part of a healthy system. And then just another example of where the collections, the preserved collections, were used for a project related on crop wild relatives. So this is mostly agricultural. So the crop wild relatives are species with genetic traits that could improve crop viability. And we've got about over 1,500 of these crop wild relatives in South Africa. Some of them are relatives of rice, of rye, of barley, of wheat, of cucumbers and tomatoes and potatoes. So a whole range of different crop wild relatives that grow in South Africa. Some of them are alien species. But what we wanted to do was to look at where the areas are in the country that are critically important for crop wild relatives, and we used collection data for that. And then also once we could map where these crop-wild relatives are, then we could collect material for gene banks. So it's often a little bit indirect, the relationship between the preserved collections and plant health. For the biobanks, it's a little bit more direct, and it's a bit easier to understand the connection. So plant germplasm collections, this is seed banks and tissue cultures, and they represent plant and crop diversity. So they're used for research in a wide range of fields. They're also used for conservation of threatened species. So the seed banks are really important for conservation and also for rehabilitation of ecosystems. So if you need to reintroduce species back into ecosystems, the seed banks are really important. And then the tissue cultures are very important for agriculture, especially as a stock for subsistence and commercial farming. So that they're used in research for the development of new varieties, or when farmers lose their existing crops to disease. So for them to be able to restart. The plant DNA bank, silica samples of dried tissue, essentially, or DNA extracts, mostly indigenous species. So these two aspects, well, I think the indigenous seed banks and the indigenous DNA plant banks have been neglected in South Africa, which is really strange given that we are known for our floral diversity. It's wealth. So we've got this incredibly unique and special plant diversity, but we haven't, we've built up good preserved collections, but not seed banks or DNA banks. And then the microbial cultures, I feel really embarrassed to talk to all these very knowledgeable people in microbiology, but I think that these cultures that are kept in the biobanks, the bacteria, viruses, and fungi, are really important, not only because they're plant pathogens, but also because they're many beneficial strains or species. So they're very important, these cultures, for research, for the commercial development of products, and then again for this reference for identification of plant pathogens and for diagnostics. So just to sort of look at how it all feeds into human health and well-being, the biorepositories are often right at the bottom, like a foundation, as a record of what we've got that's linked to agricultural productivity, protection of species, or that are used or that have important ecological roles. And all of that feeds into food security and ecological sustainability and economic impacts. And of course, you need that for human health and well-being. But often it's very hard to communicate this kind of relationship, amongst a range of people. So whether it's the funding agencies, decision makers, even other researchers, they don't often see this. And so sometimes it's useful to look at it in the other way and think, what would happen if we didn't have these collections? If we just shut them down and removed them or sent them overseas and got rid of them? What would the consequences of that be? So I think we'd have limited, unverified identifications. It would be much more difficult to understand what are the pests, what are the disease vectors, what are the potential agents that we can use to control them. We would have no verifiable locality data or records. So it's all very well to have a database, but if you ever want to check an outlier or go back, you won't have the material to go back and actually verify what it is and where it was from. The microbes change very quickly. And so something that was recorded 20 or 10 years ago might not be the same thing that's around now. But if we shut down those collections or we lose them, we can't ever go back to check what was there before. And then, of course, you would have no reserves of seeds or tissue cultures for indigenous plants or crops. And that would lead to a whole host of down the line problems. It might not be immediate, the impact, But when you start getting inaccurate identifications, you have the wrong diagnosis, the wrong treatment, the import and export restrictions could start coming in for agricultural products if we can't be 100% sure of what's there. And then, of course, we'd have no reserve stock for farmers or for conservation purposes. And food security, economy, and environment would all be impacted. So even though I think those of us who work with these collections are fully committed to protecting them, and we understand how important they are, but they still remain at risk. So the DSI funding, the funding for the infrastructure projects, doesn't cover the day-to-day operations of institutions. So it covers kind of value adds, but not the day-to-day. And because the significance of these biorepositories is not always well understood, they're at high risk of budget cuts or even closure as soon as there's financial stress. And that's not only in South Africa, but globally. And there is ongoing neglect of buildings. Often the buildings are old, they're not well maintained. And I'm not talking about UCT for the colleagues there. I'm sure your buildings were well maintained, but fire is a massive risk. And then some collections, there just isn't the capacity to look after them. And so they're at risk from various things. So some of the challenges, interestingly, are people issues. It's the very people who are there looking after the collections that can be a challenge. The communication is a big challenge. How you say what you want to say and how often you say it is really important. When you have this fragmentation where people all sort of, not all, but most of them work very much on their own without looking up and around them, it makes it very hard. And also if you work as a collective, you can help each other. But if you're all working in isolation, you're trying to solve the same problem repeatedly. There's also some challenges with trying to make sure that the research that's done doesn't align with national priorities and global priorities. And that's quite difficult. There's currently a very strong agenda driven by the number of publications rather than the impact on society. There's also some resistance to opening up access to the collections and the data. There's often a sense of ownership. I think people who've built up these collections have often climbed mountains and raised money and really struggled, and they feel a sense of ownership, and that can be difficult. There's fear that other people outside there will steal their data, steal their ideas, and so it closes up access to the collections. And we also need to look at new ways of operating for increased efficiencies and professionalizing this whole sector. And it's a change process. It does require transformation and change in thinking and operating. And then this is something that gets people really excited, are the permitting issues. I don't think we can ever talk about these collections without talking about permits. It's a nightmare for CITES or threatened or protected species. There's a whole lot of scientific registration, standing permits, collecting permits. And then there's the whole area around access and benefit sharing legislation. So all material exported needs an export permit. And I don't think most people know about that. And then also, if there's any research that's in any way linked to commercialization, it needs a special permit, and it can't be obtained retrospectively. So it means that a lot of the material sitting in these collections can't be used for any kind of bioprospecting or commercialization. And then looming, something that we're keeping an eye on quite actively, actually, is this digital sequence information. So this would be where all sequence data is treated like material, and you would have to apply to download a sequence. So, but there's all kinds of issues. There's, you know, we know that our legislation doesn't necessarily align with Nagoya. So we could still apply for all the permits here, but be blocked from publishing in Europe because we don't comply. And then, yeah, there's things are coming and going in and out the country anyway, as Prof. Wingfield said. So yeah, so we just need to be kind of clear and rational. So there's lots of work to do. There's lots of potential for these collections playing a greater role in plant health. And I always tell these communities, it's like an elephant. We're just going to chip away, bite away. And that's where I'll end. So thanks to all the institutions who participate in the NSCF and the BBSA. And of course, to the Department of Science and Innovation, I think we are very fortunate that they have recognized the importance of the collections. And thanks to all the audience and to the NSTF for the invitation. Thank you.